So today's speaker is Anna Zampetaki. She's going to talk about organoids, and I'm gonna in cardiovascular research, and I'm gonna leave it to you, Anna.
Thank you very much, Sylvia. So good afternoon, everybody, and welcome to this BHF Centre Technology Workshop on organoids as novel tools in cardiovascular research. If you look up the term cardiovascular diseases, the British Heart Foundation would describe it as an umbrella name that covers conditions affecting the heart or the circulation. And the sad reality is that cardiovascular diseases is the leading global cause of death. 3 out of 10 deaths globally are due to cardiovascular diseases. And the number is thought to reach 23 million by 2030. Low and middle income countries are actually more affected by cardiovascular diseases. And as the American Heart Association predicts, the medical cost by the year 2035 would exceed $1 trillion US dollars. So there is a lot of effort and research directed to cardiovascular diseases and generating animal models that could be used to simulate the different cardiovascular conditions is one of these fields of focus. So in here in this schematic, you can see different models from stroke cardiac remodeling to angiogenesis. This schematic is by no means complete, but a good illustration as to what different tools are available to us. Now, a typical workflow in biomedical research would include studies in cells, so 2D in vitro studies. Then promising targets would be carried over to preclinical small animal models like mice and rats. These models can be either injury models of disease or genetic models. And then the really robust preclinical data would be carried over to studies in large animals, and eventually druggable targets will enter the clinic for tests in humans. Now, the sad reality is that cardiovascular disease clinical trials have a low success rate, about 20%. And in most cases, they fail due to lack of efficacy, systemic cytotoxicity, and also unexpected side effects. So what is going wrong? Well, first of all, when we use 2D in vitro cell cultures, it's a really easy system to use. It's relatively cheap. It allows us to do genetic manipulations and genome-wide screening. But at the same time, it's a totally artificial system. So it's missing the cell-to-cell interactions, the cell-to-matrix interactions. And for all these reasons, may not be very relevant to the human biology. For the small animal models, the preclinical models, again, they are relatively easy to use. They are quite cheap to maintain, can be genetically manipulated, and we know a lot about the developmental biology of mice, so it's easier for us to understand it. But of course, they lack genetic variability. We only have a handful of different strains in mice, as opposed to the huge genetic variability that we see in humans. There are differences also in the physiology, in the immune system, and in the metabolism between mice and humans. And I am an RNA person, so I have to mention also that not all mouse homologs are human homologs genes are present in mice, especially the non-coding RNA genes. Also, there are developmental differences for some organs like the brain, and also we can detect species-specific toxicity in several cases. Now, even our most successful drugs, and here you can see the 10 highest selling drugs in the US, even the most successful drugs actually are effective only in a fraction of patients. So you see here in blue, this is a patient that is successfully targeted and helped by the medication, while in red, all these individuals actually don't benefit from the medication. And if you consider that these are the top selling drugs, then we really have a big problem. So the current paradigm in drug development is slow. The Wise Institute would state that it takes like a decade from the lab bench to the market. It's inefficient. Less than 10% of new compounds end up in the market. And it is also very expensive, about $3 billion dollars for any new compound that enters the market. So as I was saying, the European Society of Cardiology issued this call to action. And what they pointed out is that we don't really have sufficient drugs to address cardiovascular diseases. We need a new development of new approaches and the implementation of new therapies. Most of all, we need models that are more relevant to the human disease. The Wise Institute actually put it in this statement: a better way to model human biology and diseases in vitro is needed in order to accelerate the development of new drugs and personalized medicine. And this is where organoids become the center of attention. Organoids are actually human organ models that may work as the bridge between preclinical and clinical trials. So what are organoids? They are 3D tissue units. They are self-organizing, and functionally and histologically, they resemble the organ or tissue their counterpart. They can be generated from adult stem cells or induced pluripotent stem cells. Now, the European Union also saw value in this technology and invested in the Organoid Atlas initiative. So their aim is to establish a single cell reference atlas for human organoids that are derived from different organs, compare them to the primary tissue samples, both in terms of RNA-seq and imaging data. By doing so, they are hoping to align cell types between organoids and primary tissue, also infer key regulators that are aberrantly expressed in organoids and primary tissue, and interpret the results of in vitro perturbation of experiments with a mass in vivo context. So how can you generate organoids? Well, let's start with IPS, or induced pluripotent stem cells derived human organoids. Now, you can obtain iPS cells through reprogramming of somatic cells. These days, it's very easy. We have established protocols where from a few mils of peripheral blood, you can get monocytes and reprogram them to iPS. These iPS cells, left on their own devices, will actually spontaneously differentiate and give you teratomas. So what you need to do is direct the differentiation by time manipulation of environmental factors in order to get the tissue organoids. So several protocols have been established. for different organoids. And these protocols actually have 3 steps. So in the first step, you actually establish the regional identity, the embryonic layer that you direct the differentiation, endoderm, mesoderm, or ectoderm. Then you terminally differentiate to the desired cell type. So you have, for example, mesoderm to vascular cells. And finally, you provide a 3D scaffold so that your cells can expand and form tissue cultures, tissue structures in a 3D form. Now, the great advantage of induced pluripotent stem cells is the fact that they can reflect the genetic heterogeneity that exists in the human population. But apart from that, they also have an additional benefit, and that is that they maintain epigenetic memory. So several groups now have reported that the cells that are derived from a disease condition like diabetes, for example, continue to behave as diabetic cells, even if they are cultured under normal conditions. So this is an additional advantage, and patient-derived organoids can be employed for disease modeling. We can also do drug testing, and they are amenable to gene editing to elucidate the mechanisms of disease. Now, I'm going to talk about a bit more about human blood vessel organoids and derived from iPS cells. This protocol also has 3 steps, the mesoderm differentiation, then vascular differentiation, and embedding into a substrate so that you have vessel sprouting, vascular networks, and blood vessel organoids. So just to put some images on all these steps, we start with iPS cells. Then you have the aggregates that will differentiate to mesoderm and vascular progenitors, as you can see here. After embedding into the substrate, you have the vascular network formation that is extracted and placed into 96 well plates. What you see here are actually different organoids. Finally, on day 15, you have the blood vessel organoid. If you want to characterize the cell composition, you will find that here in blue, you see a lot of CD31 endothelial cells, and also positive cells for PTGF-beta, so neural cells. The size of the diameter of the vessels that we get points out to capillary formation. These capillary structures, as shown here, are part of the microvasculature. So the blood vessel organoids actually recapitulate the microvasculature. Of course, there are also some smooth muscle actin positive cells, but the majority of them are pericytes that are in close contact of CD31 endothelial cells. Now, here I just included a movie just to show you how dense the vascular network in these blood vessel organoids is. And when transplanted in vivo in nude mice, they become part of the circulation. So you see that the vessels that they formed are well perfused. You can detect arterial venules and venules formation in vivo. Now, this system was used to model diabetic microvasculopathy. So diabetes has a very severe effect in the microvasculature. As you can see here, these are microvessels from diabetic patients. You actually have increased deposition of extracellular matrix of collagen 4 and laminin, so that the basal membrane is thickened. And this thickening actually leads to a stiffening of the vascular of the vessels in diabetic individuals. And importantly, when in vitro vascular blood vessel organoids, when exposed to hyperglycemia or diabetic conditions, they also show this thickening of the position of thickening, the position of collagen 4 and thickening of the extracellular matrix. If you get these vascular blood vessel organoids and you transplant them in vivo in mice and then induce diabetes in mice, you again are able to observe the thickening of the extracellular matrix of the basal membrane, indicating that these blood vessel organoids actually behave as a microvascular tissue. Now, I'm not gonna go into all details about the experiments that the authors did to figure out the mechanism of this membrane thickening, but I'll tell you that following drug screening, they identified NOTCH signaling as important in this regulation, and then went on to do gene editing and generate edited blood vessel organoids. So they started with iPS cells, did CRISPR-Cas9 gene editing, generated DLL4 and NOTCH3 knockout lines, and from these lines they generated knockout organoids as well. So first of all, they confirmed that their gene editing worked. And then used these organoids to culture them under non-diabetic and diabetic conditions, as expected, under control. Using control organoids, you have a huge deposition of collagen 4, but if you knock out DLL4 or NOTCH3, this response is no longer there, indicating that gene editing of the blood vessel organoids can also be used to interrogate, to understand the mechanisms involved in the pathology. Now, a modified version of this protocol was also recently published, and these authors here provide a protocol where you can also use inducible differentiation of embryonic stem cells to include neurons in your vascular organoids or skeletal muscle cells. So they end up with Neuro-Vascular and Myo-Vascular organoids, and the way to do it is by using inducible transcription factors for either neurons or skeletal muscle cells, as shown here. Following staining, they were able to see that indeed you have both lineages in the neurovascular organoids, as also confirmed by single cell sequencing. And also in the myovascular organoids, following electrical stimulation, you see the profile of gene expression that fits with the presence of skeletal muscle cells in these organoids. Now, a question that comes to mind after all this is whether there is an added value of using induced pluripotent stem cells-derived organoids versus induced pluripotent stem cells-derived cell lines. And the truth of the matter is that there is a very important difference. So here, iPS cells were used to generate endothelial cells or vascular smooth muscle cells, but on its own, each line cannot actually generate the deposition of collagen 4 in diabetic media or hyperglycemia that are seen in the organoids, in the blood vessel organoids. So it is an indication that actually the 3D structure and the cell-to-cell interaction, the interaction with the extracellular matrix as well, is important and recapitulates a critical figure that exists in the microvasculature in diabetic patients, and therefore is able to reproduce this phenotype. Now, the second example that I would like to mention is the screening of compounds that have a pro-proliferative effect in cardiomyocytes. And in here, the authors actually tested more than 5,000 compounds in iPS cardiomyocytes versus fibroblasts, identified 100 compounds that are inducing proliferation in cardiomyocytes without affecting their electrical function, and then took these compounds over to the cardiac organoids. So what is interesting is that out of 100, only 10 were actually effective in cardiac organoids. And when they matured these cardiac organoids further using maturation medium that induces a metabolic switch, only 3 compounds were actually confirmed to have a proliferative effect in the cardiac organoid. So again, it suggests that the 3D tissue structure captures a part of the biology that is missing in the 2D systems, even if we are using a human model. So moving on to heart organoids, this is from a very recent publication. I think it's by far the most advanced protocol so far. You have 3 steps of a differentiation. Again, we start with embryoid body formation, moving on to mesoderm and cardiac mesoderm formation, and finally organoid development. You can see here the timeline for day 0 to day 15, how the phenotypically the organoid changes as well, and also how you get an increase in cardiac troponin T deposition as the days progress. Apart from cardiomyocytes, you also see cardiac fibroblasts being present in the organoid, also endocardial tissue and endothelial cells. When they look at the percentage and the different lineages that they have in these heart organoids, they actually discover that it resembles very much the fetal heart, with the majority of cells being cardiomyocytes, but also cardiac fibroblasts, and also endocardial cells, epicardial cells, and endothelial cells. And this is how the structures would actually look like based on their analysis. Just to show you some more data of the functional and structural features of the organoids that actually resemble the developing heart. So you have the sarcomere here, T-tubes here, again the structure point to the developing heart, and so do the electrophysiology recordings. I thought it would be nice also to show you some of the movies that they provide on the beating organoids and how they resemble the heart. Here you can actually see a staining of the organoid with India ink, so that you, under light microscope, you are able to see a central chamber, something that would suggest that you have a heart field formation, and also adjacent you have beating parts. And here you can have a 3D reconstruction of the topology that suggests a high degree of interconnectivity between 4 or 6 chambers near the center of the organoid. Now, the other thing that the authors did was actually generate some iPS cells that carry a calcium reporter, and what you see here are calcium waves, which are typical of cardiac muscle. So again, some functional data to indicate that this is a good representation of cardiac tissue. Finally, they also show that you have some endothelial cells in there, so a vascularization of these organoids is also important if you want to maintain them for a substantial amount of time in vitro. Having said that, this model generates organoids that resemble the fetal heart, the authors thought that a good way of using this model would be by investigating the effect of media that would sort of simulate the pre-gestational diabetes state. So what they wanted to see was congenital heart defects as a result of this diabetic medium in the organoids. And as you can see, even by looking at them, there are big differences in their morphology. Your diabetic organoids have a form of macrosomia that may indicate that you have cardiac hypertrophy, just like you see in the congenital heart defects. Also, they studied the metabolism in these organoids, and they showed differences that are in line with what we would expect in congenital heart defects. And as far as the electrical activity is concerned, you see that there is some indication of arrhythmias, suggesting that this is a model that could be used to study congenital heart defects. Now, we're going to change gears, and we're going to talk about the adult stem cells derived organoids. And everything started when the Hans Cleaver's lab discovered that there is actually an LGR5 positive cell type in the intestine that can produce intestinal organoids. So you can see here at different days of differentiation, and finally the intestinal organoids. Effectively, these cells can produce micro crypts with microvilli. And this mini-gut culture system that is shown here would involve isolation of these cells, then differentiation to organoids, then they can be applied to an injured intestine where they will form a new epithelial layer with the help of Wnt5 positive cells. Now, there are several basic and clinical applications for these mini-gut organoids. They can be used as experimental tools, diagnostic tools, and as therapeutic tools. The remarkable thing about it is that you have almost 100% success efficiency in establishing the cultures from individual patient samples, and therefore you can have live biobanks, and you can actually differentiate these tissues in different organoids. Again, here the protocols differ somehow, but depending on the tissue origin, these cells can actually give you the respective tissue organoid. Now, the biobank that I was referring to already exists, and it is the Habtrecht Organoid Biobank. What they do is have patient-derived biopsies, and these then are differentiated to organoids. So they are all derived from adult stem cells. They are physiologically relevant. They are also genetically and phenotypically stable, clinically relevant, and can be used for large scale screens, and they are suitable for genetic manipulation, and also they can be amenable for in vivo modeling. So all this application, in vitro disease modeling, drug discovery and development, clinical trials in this, companion diagnostics, effectively generating a list of biomarkers associated with a specific disease, safety and toxicity, and personalized medicine are part of what these collections can be used for. I would like also to mention a few things about cancer organoids, and again, methods exist now to generate cancer organoid biobanks. As you can see here, we can have normal tissue organoids and organoids derived from tumor tissue. You can also manipulate the normal tissue organoids to generate tumor organoids. And this approach really captures the inter-patient variability. It can also recapitulate the drug response to the of the corresponding patient, and also can be used for the development of biomarkers predictive of drug response. So this is a personalized approach. You can have tumor organoids derived from an individual, then use them for testing hundreds and hundreds of different compounds. So it is what is called N=1 clinical trial. The compound that is more effective then can be used to treat the patient in a more efficient manner. So here I'm giving you an example of rectal cancer organoid. So on the top you have the tumor of an individual, and the organoids that were derived from this individual, staining for various markers of cancer. And when the authors did a study using a cohort of patients, they found that actually there is a good correspondence of the way that in the clinic patients respond and organoids respond in vitro. So in here in blue, you see patients that had a good response to the therapy. And as you can see, the organoids that are derived from these patients are sensitive. So you have you have elimination of the cancer, which is what we are actually looking for. There were also individuals in the study that had a poor response to the treatment, and again here their organoids so that they were resistant. So again there was a good correspondence between what the organoids saw and what the actual patient, how the actual patient responded. And unfortunately there is still 20% here with good or poor response that the organoids derived from these individuals showed there was no corresponding response between the organoids and the patient. One more example that I think is remarkable is the generation of endo and ecto cervical organoids. Here again, the staining shows you how close to the actual normal tissue, healthy tissue in this case, these organoids are, so that it can be used to study the biology of cervical epithelium infections and also cancer. And in here here actually you see some cancers and tumor organoids. And the remarkable thing about it is that all these are actually derived from material collected with a pap brush. So this approach actually eliminates the need to have solid tumor biopsies, and hopefully will make things easier and provide a more personalized response for an individual's response to medication. Now, several corrections are already available, and you can see that you have over 100 corrections where you have normal tissue and tumor tissue from the same individual. These can be licensed and used for drug screening. You have the capacity to do 384-well formats or a high throughput screening for these organoids to identify toxicity and identify compounds that could target the cancer tissue. There is also the Human Cancer Models Initiative, and the Wellcome Sanger Institute from the UK is also part of this initiative. In this case, again, we have a vast collection of cancer cell lines and organoid model. They issue what they call cell model passports, where you actually have genetic information and at the same time a phenotypic characterization. So all that will hopefully lead to better screening and more personalized approach to medication. Now, there's no escaping it that when you are when you are treating a patient with a drug, apart from the intended tissue target, you also affect other organs in the body. So drug absorption, distribution, metabolism, and excretion would affect organs like the gut, the kidneys, the liver, the heart, and they are all they can be also very sensitive to this medication. So the liver in particular is an organ that was shown to display very high species differences in this ADME processing metabolism of medication, and therefore we will need a human model to study the pharmacokinetics and pharmacodynamics of drugs in vitro. And in this case, people came up with the idea of multi-organs on chip systems, or having a sort of different organ models connected and under the same circulatory system, so you can have a human on a chip. Now, this technology relies on a different platform, the organs on chip. This is again a human organ model. So what you will have here, you can see it is two microfluidic chambers, shown one in yellow and one in blue. They are on a flexible polymer. Usually it's the size of a USB memory stick. On one chamber, you have endothelial cells, and in the other, you have epithelium of the tissue that you want to target. In between, you have a membrane that also has pores, and it can be stretchable. And the whole system can really be very closely monitored. So there are several sensors that control everything from flow to electrical stimulation, mechanical stimulation, even dimensional control. Everything can be very closely monitored. So far, very advanced chips have been generated for brain organ on chip, for example, where you have the vascular network and the brain network shown here. So the vascular network is consisting of endothelial cells, but the brain channel actually has several cell types like microglia, astrocytes, neurons, pericytes. So it is quite a good representation of at least part of this specific region of the brain. And as you can see here, the staining, you can see the monolayer in both parts of the membrane. This system was actually used and they managed to reproduce several aspects of Parkinson's disease. And also you can have a airway, human airway on a chip where you have the epithelial cells in one chamber and the endothelial cells with circulating immune cells, neutrophils in that case. On the other side, this system was used to repurpose drugs that could be effective against COVID-19. And again, you see that they managed to get very nice monolayers of cells in both sides of the membrane, a stressable membrane that can actually mimic the breathing motion as well. By far the most advanced for platform so far was this multi-vascularized system that was reported by the WISE Institute. So what they did, they used 8 different organs on chip connected into one system in what they call a human body on chip. So they have the brain, the lung, the skin, the kidney, gut, liver, heart, and the brain blood barrier with a substitute of blood circulating throughout the different organs like this. You can see here also very nicely they managed to maintain these organs on chip for 3 weeks and used labeled insulin to detect accumulation in different organs. It is encouraging that actually you do have accumulation at levels that resemble what we know from patient data. So it managed to capture some of the pharmacokinetics that exist in humans as well. The second system, it is a bit more simple, but the principle is the same. So again, vascularized organ chips for bone marrow, liver, kidney, and all of them are connected together. They have also shown that there is the cisplatin accumulation follows the pharmacodynamics that we know so far from patient data. So I think in conclusion, we can say that organoids and organs on chip can recapitulate the human physiology and heterogeneity and may facilitate a more effective drug development pipeline and precision medicine approaches. But of course, there are several challenges ahead and there are lots that need to be done. So first of all, we need to standardize the protocol so that we have reproducible organoids in terms of size, in terms of cell composition, in terms of function. We also need to eliminate the use of animal products like the matricel, for example, because it is unknown composition. So something that is more defined is essential if we are to carry any of these finally in vivo. these, or most of these organoids actually don't have shear stress or media flow. So this is another element of physiology that would be critical and is missing at the moment. We should also improve the maturity of the derived tissues and establish protocols that move closer to the adult tissue and not so much the fetal tissue, and also generate multi-lineage organoids. Some of this is already underway, but of course a lot more need to be done, because our organoids should definitely have vasculature, immune cells, and innervation if possible. So finally, I would like to thank the British Heart Foundation and the BHF Centre for funding our research, and I will be happy to answer any questions.